So fantastic. We have Kelsey Bison is joining us. Kelsey is at NASA headquarters, and she is going to tell us today about biodiversity from PACE and is also going to join us in person, you said tomorrow evening.
Okay, thank you. Thank you so much. And I'm really excited to see the room, even though it's on my screen. I'm so sorry that I couldn't be there this morning, but I'm really looking forward to meeting and spending time with everybody later this week. So I was thinking about, what I wanted to put together for this talk, and I initially was hoping to share some results from the ecological modeling that I did, but then I was sort of thinking it wasn't the most exciting or relevant for this group. And so I wanted to talk today instead a bit about challenges and possible ways that we can overcome them in addressing biodiversity using the PACE mission, and plant some seeds for ideas of things that could be worked on perhaps this Hack Week or in the future as you're considering other projects and side projects and so forth. So I've been enjoying seeing, and I know as you have as well, just extraordinary images from PACE since it launched about a year ago. And when I think about these in the context of probably everything that people are experiencing this year, it reminds me that life is really beautiful and that we're living in a Renaissance period in terms of Earth satellite missions. Both of those things may not necessarily feel true. And so I wanted to focus my talk sort of around those themes today. And to go back and really with some appreciation for life on Earth. And, not to be dramatic, but, 13.8 billion years before PACE launched, we didn't have incredible biodiversity on Earth, of course. And we've made a lot of advances in our ability to understand the structuring of life on Earth, starting really at the subatomic level. And so we know, for example, that there's 12 fundamental subatomic particles of matter. These are quarks and leptons, and they have, each of them have 6 different characteristics. Sorry, yes? Yes. No, I appreciate it. Thank you so much. That would have been frustrating. Would have been there the whole way. Okay. And those 12 subatomic particles of matter structure and have given us, 118 elements in the periodic table. Can I get us also a check? Are you seeing Teams messages come in? I wonder how to hide those. It might be pretty noisy the rest of the talk. I get, does anyone know who's been a NASA person who on Google Meet if there's an easy way of silencing? That might work. I don't know if it will fully fix it, but yeah, let's try. Okay, perfect. Okay, and you can see the full screen again? Okay, amazing. So, yeah, I mean, so we have, roughly 100 elements in Earth naturally. Those create around 6,000 unique minerals. Again, all, inorganic structures. And then from that, we have a whopping estimated 10 million species on Earth. And so the scaling here is obviously very nonlinear, but I just wanted to take a moment because it really is extraordinary to think about how we have such a diversity of life on Earth and what supports us in our oceans and our ecosystems, not to mention, habitats on land, et cetera. There are some studies that estimate that of the species that we suspect we have on Earth, I think there's something like 2.7 million identified, but up to 10 million. We have most of that, the vast majority of that is undiscovered in the ocean. I'm sorry, I don't think closing Teams helped, but in any case, it will just, yeah, we'll go with it. Okay, so I hopefully you can hear me okay. I wanted to hear from you and from the room why you think we need to quantify and describe changes in biodiversity. Like how is it helpful thinking about the structuring of life? Unfortunately, I can't see anybody, so I don't know if there's hands, but you could just shout it out. No. I'm so sorry.
Figuring out the system too. Do you hear me when I'm using the mic?
I can hear you beautifully, Ian.
Oh, okay. I'm going to pass the mic around.
Yeah, rhetorical, but so that we don't lose it all.
So that we don't lose it all.
Yeah, that's Chintan's answer. He said it
Very
was rhetorical,
true.
but it's very true.
It's a good point. Yeah. We can't protect and understand what we don't know is there.
If we're trying to understand the Earth's climate, the biodiversity, I'm thinking about the ocean and the way the biodiversity in the ocean is such a huge driver of changes in like carbon dioxide. So, but also in the Earth's biodiversity, also a huge driver. So just kind of understanding what's happening at a global scale with our climate, we need to. It's essential to understand the biodiversity.
Absolutely. Yeah, we're a living planet, and just as I, started by saying we scale from subatomic particles up to, an extraordinary amount of life on Earth, it's not a one-way street, and so the amount of thriving or not life, it influences all the other aspects as well, and we really are truly a living planet.
Yeah, I think the global warming has affected mostly the marine biodiversity, coral reef, causing ocean acidification. That is affecting most of biological species. So I think we need to quantify in order to know well what will happen, to know the rate of loss of these species. This will help to maybe provide some solution for how to protect our biodiversity.
Thank you. Yeah, it sounded a bit mumbled on my end near the end, but we absolutely need, if I hopefully heard it correctly, ways of understanding the different species and ways of observing them, across regional and global scales. Are there other hands or other ones before I move on? Sorry, I can't see anybody.
One more, it looks like.
Okay, thank you.
I think of the idea of One Health. So the idea that a healthy environment, healthy people, healthy animals, we are all intertwined, and maybe more ways that people may think.
Mm-hmm. Absolutely. I was just reading the other day about the link between, fisheries in the North Atlantic and national security, and thinking about wars that have been fought over food security and understanding national fish and so forth. And it's a huge issue. Thank you all so much. So I set up sorry, was there another thought? Okay, thank you. Sorry, I was thinking, I heard some lingering sounds. I also wanted to talk about challenges that we have now with more of an eye specific to space-based sensors and quantifying biodiversity. And I'd love to hear from those in the room what your thoughts are on this. Keeping in mind, when we're thinking about biodiversity, we're not just thinking about the phytoplankton that we know and we love dearly, but also other trophic levels and other aspects, both in the ocean and on land.
Just one thought that we actually had a small discussion over breakfast amongst myself as a cloud scientist and people who see clouds as a nuisance, that sometimes you can't always observe the places that you want to see. So whether because you have some interference in the atmosphere or because there's some life that lives deep in the ocean that you wouldn't see from a space-based sensor.
Mm-hmm. Yeah, absolutely. There's fundamental constraints physically on what we can see, especially using the passive ocean color technique. I can't tell you how many times there was a really interesting Earth event, and I went to see if there was satellite data available, but it was too cloudy, or it happened, at a time that was too far away from our overpasses.
Another challenge might be distinguishing between species. The same species of phytoplankton or, vegetation may look very similar, so you may sense that there is a type of phytoplankton, but that doesn't tell you if there's diversity present.
Mm-hmm. Yeah, absolutely. That's a huge challenge.
I was just going to mention, actually, quantifying what biodiversity is. If you're out doing like a fisheries survey, there are some creatures who can avoid nets better than others, right? So you're not always just the sampling mechanism alone of trying to like detect biodiversity is can be inherently flawed. So just trying to get that actual background information is difficult enough as it is.
Yes.
Yeah.
It is really challenging.
Yeah,
Are
and-
there other hands? Okay, great. Thank you.
Yeah, another challenge is can be data collection. Our ocean is large. We have different ocean in different regions of the world, and then collecting this data is very, very hard work to do, and sometimes there are some missing of instrument or captor that can be used to capture to measure the data. So that's why we have, for example, we use, for example, data from satellite that can maybe cover different regions of the world. Yeah.
Sorry, what was the last part, the last few words?
It was, that's why we need to use satellites to cover different regions of the world at a large
Yes.
extent.
Mm-hmm. Yeah, absolutely. For as many challenges, I think, as there are quantifying biodiversity from space, there's equally as many opportunities, not to be corny, but I think we're sitting on a gold mine with PACE data. Are there other hands?
No, I think we're good.
Okay, thank you, and thank you so much for facilitating that, Ian. So there were 5 or so kind of main themes I was thinking about in putting this together. There's certainly more, but I think these 5 are definitely achievable to overcome within the next few years, and it'll be really exciting to see advances in this area. And so I wanted to walk through some of the challenges as I see them and hear feedback from others on what they've experienced when they're working with these sorts of data products and thinking about the questions that are achievable for us to even ask using these types of data products, and then those that are just outside our reach, but could be addressed with combination of methods. So one of them that I think about a lot, especially in some of the wildfire research I do, is how uncertainties propagate as we derive more ocean properties. And so with the passive color technique, we've had nearly unlimited, but some limitations, abilities to look at life on Earth. One of those limitations is that the signal is at the top of the atmosphere. And so over 90% of our signal, as you know, is the atmosphere, which is great, especially great for atmospheric scientists. But when we really want to understand the ocean contributions, we're working with lower signal and need a bit more creativity there to pull out data that we can explore, in depth. This is a beautiful challenge to have, but our ocean is incredibly complex also. And there's a number of species interactions, and not to mention species we don't even know exist, right? That are out there influencing the ocean at different times and in ways that we don't fully understand, nor have the observational techniques to fully understand. And so that's a level of uncertainty in the system that's a natural one, but one that is both, I think, inspiring, but also, of course, a challenge. I think this was mentioned momentarily ago about how ocean color technique, it doesn't work when there's a lot of clouds. It's also not depth integrated. And so we have surface information primarily, and we know the ocean is a very deep place and that there's, life all the way at its depths. And these are not necessarily constraints in itself, but if used in isolation, we would have a limited view of the ocean in terms of the full water column. And another limitation, which is more on us, but I think it's always good to remind ourselves that we're learning and there are ways that we can build models, especially mechanistic models, that are only as good as our conceptions of the ecosystem. And so how we choose to parameterize models, what we allow to be varying versus what we assume to be low variability, those are all at some point assumptions that can be tested and can hold up, and sometimes maybe they don't. And so this is another consideration when thinking about how we can stretch data to work for us within a modeling format. And then, especially thinking about the remote sensing approach, we have an amazing vantage point and a synoptic one. At the same time, depending on how we're using these data, we might treat them as independent when in fact there might be some time varying component. And if we're looking at, for example, one picture here, it doesn't tell the whole story. We don't know where that water mass has been, and how the water has evolved through time and what that means about the phytoplankton communities, the zooplankton, the fish, et cetera, that are contained implicitly in this image. So I wanted to walk through all of these 5, give a few examples, but then also talk a bit about how I think we can overcome these with PACE data and with other types of data products as well. And so the first one, as I mentioned, is that we are working with top of the atmosphere radiance data that are derived into spectral remote sensing reflectances. You can see here that in this sort of conception, this is from a paper by Dave Siegel from a few years ago. As you go further down, and particularly into PCC, phytoplankton community composition, or products that can help quantify even further from that, net parent production, you accumulate uncertainty. So by the time you get to the food web modeling stage or empirical models, you have a lot more assumptions and uncertainty that's in your system, which is totally fine, especially if you know what it is. But it also does create some challenges in how well we can answer these problems. One example of this is from some recent work identifying seasonal biases in remote sensing reflectance relative to in situ data, and this is at the Moby site in Aqua. Here we're showing the inverted values, and so the annual cycle, the months are on the x-axis, and you're looking at the ratio on the y-axis between Modus Aqua data and Moby. And you can see some products more than others are affected. And so chlorophyll, for example, in the green dashed line, is it has these biases, but it maybe 20% of the variability, whereas CDOM, or the absorption of dissolved matter in blue, it's much closer to the one-to-one line. It's less affected by this anomaly, but then backscattering, for example, can be also quite high, up to 30% or so, and that is directly empirically used to quantify phytoplankton carbon, for example. And we know how chlorophyll has been used a number of ways. And so these are some examples of how we might see propagation of error show up in data that we're using. And I wanted to plug Riley's paper, which was just accepted on Friday. And so stay tuned for that. It talks a bit more about some of the periodicity that we're observing in satellite ocean color observations, with ideas about how we might address that. On point 2, as I said, I think this is more of a beautiful thing necessarily than a challenge, but still it is really challenging, right? We have an incredibly complex ocean with, size ranges, 6 orders of magnitude. We have processes that are operating across seconds to those that are operating across millennia. And we have a set of tools to use. Not necessarily are they all interoperable. And at the same time, it's extremely expensive, both time and cost, to go and collect these types of data at sea, which give us highest taxonomic resolution, things like genomic data or image data from the water samples, et cetera. So this is certainly a challenge, but then in addition to that sort of complexity, there's complexity from an evolutionary perspective. And so I wanted to highlight some of Sasha's work here. And this is on the left, a table reprinted from a textbook a bit about pigments and pigment sharing across different lineages. And, wouldn't it be nice if we had unique, that dark gray box everywhere that we're looking at phytoplankton? But that's not the case. And so there are a lot of challenges associated with understanding how we can best use, pigment data or, spectral absorption data to best discriminate phytoplankton groups from each other. And on the right-hand side, we're looking at an analysis that she ran looking at correlations where the gray bars represent significant correlation statistically between different pigment types spectrally across the different phytoplankton groups. And this is using a derivative analysis technique of the spectral remote sensing reflectance. And really what I wanted to focus on here is just to illustrate that there's a lot of variability. You know, there are times where there might be features that one could identify different parts in the spectrum that are stronger and more strongly correlated and could be attributed to different types of phytoplankton versus times where it doesn't offer as much use. And so we are working with an era of more information than we've had before from our satellites, which is incredible. Not only do we have hyperspectral capabilities, but also UV and polarimetry, and understanding how best to use that will be really important as we move forward. The other, aspect that we've talked about is that ocean color is really looking at daytime values in the ocean and, with a signal that's exponentially weighted to the upper few meters. And we know that phytoplankton, they attract many species, not just us, they also attract zooplankton. And these are the biggest migration on Earth, it's said to be, and it happens every day in the ocean. And so we have, these animals that are hiding at depth during the daytime to escape predation and the darkness, and then at nighttime when it's safe, they come up to feed. It's a really incredible process, and it oftentimes will bring up a lot of other predators as well. I was in Ohio with my family last week watching Shark Week with my dad, and I was learning a lot about shark behavior and different types of fish off the coast of South Africa, and they had a strong diel cycle as well. And so this is really a big forcing on the ecosystem. But it is challenging to observe from passive ocean color, which means we're not getting the full picture of biodiversity unless we think about other processes that we can account for at different times of the day. This has been pursued a bit by LiDAR data, but I think that it's ripe and there's a lot of additional analyses that could be done. So it's example of, in this case, we're looking at field data. The x-axis is looking at different days sequentially, and the y is looking at the particulate backscattering coefficient. And you can see that, there's a baseline during the day, and then you get these big spikes at nighttime, which is in the gray shading. And at first, if you were working on the instrumentation, you might think, oh no, something's wrong. But really, these spikes are the treasure here. And thinking about how we can best use those and incorporate them into understanding the types of animals that are coming up across different size groupings, phenologies, et cetera, will unlock, I think, a lot of our understanding in what we can do. As I mentioned before, and this is not a dig on any model, we need to think about how we can develop better mechanistic models and also statistical ones, which incorporates, AI or machine learning when we know best how to use it. But within our mechanistic framework, we want them to be complex enough that they're actually useful. But at the same time, if it's overly prescribed, there's a lot more room for error. And of course, we're coming up at the limits of our own understanding when we start to over-parameterize and think about why do we are doing now, how certain phytoplankton groups are grazed upon, and then how those zooplankton are grazed upon from higher trophic levels, and how those things are, scale across different physical disturbances in the water column, nutrient availability, ice conditions. Like, you can think about these across a giant phase space. And it's clear that, we're able to make progress with simple models, but it's good to keep in mind how they may or may not be, working well. And so one of these are areas that I've been exploring a lot recently and trying to understand how we can better match our observations in life with what we see in models. And, you may know this from your own work, like, you go out into the middle of the ocean, if you filter enough water, you'll start pulling up bigger particles. No, they're not in high concentration, but they're not, fully extinct. And some of the models that we use currently, incorporate parameters that might cause extinction prematurely. And so, this was initially a really big question where we thought we'd have a profound answer. And it turned out it was not necessarily profound, but, it was a parameterization of a linear mortality term that created complete differences in the size structuring of communities. And what was essentially happening is that, as you see in these basic sort of NPZ type equations here, we can look at the change in phytoplankton over time as the growth rate minus the lost terms by grazing. Typically, there might be a linear mortality term. And if you add a linear mortality term, essentially what it does is it kills off anything that's growing slower than the mortality term that is assigned, versus a nonlinear mortality term, which might be more realistic in nature in terms of thinking about how phytoplankton die. I mean, do they die of old age? Like, these are all questions that, when thinking about modeling, become really significant in adding. And I've certainly, recently even incorporated parameters at times because they've been used widely in the literature, and I thought it made sense. And then when I really worked at it, I couldn't totally explain and justify the use of these things. And so I think additional exploration with simple modeling frameworks and also parameterizations can help us understand how we can or cannot preserve diversity in models relative to what we see in observations when we go to sea. The other challenge that I was thinking about is that we're working in a system where satellite data are offering really powerful snapshots in time. But we know that different groups be successful at different times. And so you can think about this as phytoplankton, you could think about it as zooplankton, you can think about it as fish, whatever suits. But basically here, I was running a model where we started with equal distribution, a uniform distribution of 4 different species. And on the top, we didn't allow immigration. So we were basically saying these are separate areas of the ocean that don't interact. And then if you run that out, up to 100 generations, you can see that for those, we pre-assigned these species certain characteristics that made them more fit than others. And so in the top right, for example, the blue species 1 had a slight edge over the others. And what I mean here is really it was a slight edge. I think we assigned a growth rate that was, 1 or 2% higher than the other ones. But over time, that selects for that species to outperform the others. And we could see that in nature, but we know the ocean is extremely physical environment. It's highly dynamic. And so when you start to add immigration and you allow species to move, it conserves all species, even when you go out to higher generations. And while this is an extremely simple sort of game model, essentially, it also allows us to understand how we might conceive of incorporating different types of data, that when we look at a satellite image, for example, we can think both about stable biodiversity, which would be the type of biodiversity of equal fitness organisms that are living there and able to thrive. And then this unstable component, which is maybe physically derived or from other reasons. It could be, a flux of nutrients from an atmospheric deposition case, that does slightly temporarily adjust the phytoplankton community, but doesn't necessarily imply that all of those species are well suited to be forever. And I like this as a lens because we can think both about the concept of invasive species or not, and ecosystem resilience, and thinking about, answering certain questions. How biodiverse do we need to be to have stronger resilience? In some cases, this top plot, it might be fine as long as these species have the ability to flex with their environment. But if they don't, for example, then it would be really problematic, versus the bottom plot, which has, obviously a lot more biodiversity at the end of the run. So I think I have a few minutes left, but I wanted to go over some of within each of these challenges, areas of opportunity that I think could be really fun to work on and would be great hack week sort of projects or hack week sort of conceptions for those of you that hopefully make a lot of friends this week and think about ways in which you want to work on things together beyond hack week. And so while I did mention that uncertainties are, they propagate as you derive ocean properties more, we can minimize them by working together across sensors. And so PACE is an amazing platform because it has, these 3 instruments that offer us just so much. So there are, new ways that we can think about atmospheric composition with PACE that can allow us to directly include these sorts of data products in our atmospheric correction and then better produce ocean data from that. So this is showing some of the variability of that, from cloud fraction to aerosol depth, single scattering albedo, even, black carbon and brown carbon, which will change the game in how we can identify wildfire volcanic events and how carbon's transformed, as well as the size of particles as well. there's not so much that we can do about the complexity of the ocean. Let's hope it doesn't become more simple. At the same time, we can use a lot of data streams in tandem to eliminate blind spots. And this is not just a satellite conception, although I said I really think technologically speaking that we're in a renaissance right now. And I try to think about that a lot when it doesn't feel like we are in other areas. But we have a lot of tools to use. And so across global scales, not only do we have a lot of satellite observations, but we also have, the Argo fleet, and we have cruises of opportunity, and aspects like the Tarak expedition that is given global data, not necessarily synoptically, but providing water column data that can, allow us to address, incorporating genomic data or acoustic data with PACE, and asking these questions that will start off extremely exploratory, but we can finesse as we continue to evolve. This is an example that's a bit more, it's higher level and less useful for biodiversity perhaps, but one in which it is using two satellite data types together. And so, thinking about there are so many advantages with the passive technique. One advantage with the active sensing technique, LiDAR, light detection and ranging, is that it's less influenced by the overlying atmosphere. And so it can provide slightly better backscattering data products. And so in this case, this very simple case, we were working with the generalized inherent optical properties algorithm, and we modified that to directly ingest LiDAR or simulated backscattering data. And so that's this path 2 on the schematic here. And then we produced absorption values that in simulated runs had higher performance. Globally, it wasn't possible to test this because of the sparsity of the data products. But I think this is a very, very simple case. I think we could expand this a lot and use the GIOP as a jumping off place to think about adding, for example, physical data products, or making a Bayesian scheme where we can add additional data streams that both help us improve our degrees of freedom, but also constrain things by informing it with realistic data, and that could be static products that we include, or it could be dynamic ones. So we could ask questions about, as the water mass evolves, we have time derivative information about the water column. What might that imply about the phytoplankton community or the CDOM situation, et cetera. So, I mentioned, yes, we have challenges with the surface integration, but we also do have this global fleet of Argo floats. We have a lot of regional LiDAR data. We have LiDAR everywhere globally. Right now, we only have one LiDAR flying that can be used for ocean data, which is ISAT-2. But EarthCare, while not intended for ocean data, it might be possible to extract something from that. So stay tuned. There are a lot of areas that I think we could work with LiDAR data, even though we don't have an ocean optimized LiDAR in orbit yet. And then, of course, there's a lot of optical data that's coming on board from underwater vision profilers, the image flow cytobot, and then we can have additional data, Niskin bottles capturing pigment data. Not to mention, right now there's a giant boom in eDNA that people are really excited about. I think we need to think more about how are those data quantitative, and it can and to what extent, and how and under what conditions, et cetera. Can we link with PACE data and expand our understanding? But there's a wealth of data that we have here, and thinking best about how we can incorporate them, I think will be really fun, but also extremely meaningful about how we can advance our understanding of ecosystem. And so on the left, I wanted to show some examples of what LiDAR data can give us. This is the bottom 3 plots are over the ocean and go around to 30 meters depth or so. And we're looking at on the top backscattering KD, and then at the bottom the particulate depolarization ratio. And you can see some spatial differences here, but moreover, I wanted to sort of advertise this as it's sort of, we can think of it as like an Argo float essentially, but it's has a lot better spatial coverage. And so as we advance this technique, we'll have a much better, informed view of how phytoplankton or particles are structured in the ocean. And with these types of data, if we get them globally every day, eventually, we'll be able to maybe even quantify particle sinking rates from space, which I get really excited when I think about these questions that we could potentially answer. On the right, I'm showing data from Niels Hengens from 2020. This is also using Argo data, and so there's Argo data plots on the left, looking at temperature, chlorophyll fluorescence, backscattering, fluorescent dome, and some. And then on the right-hand side, he's showing groupings of copepods, crustaceans, rhizaria, and you can see that there's links between some of the spiking and different groups and where they are at depth in the ocean. And so improving our understanding again of across trophic levels and with optic data, I think that there's so much there that are ripe for new discoveries. To some extent, these are new tools, even though we've been using them for a while, but approaching them with a new way, I think, tends to generate new discoveries. And this is an area that is really exciting. We are limited still by our understanding that will never not be true, but at the same time, there are some new techniques that can help, that are really exploratory. And that's both a strength and a weakness. It's difficult to interpret and understand in many cases why AI and machine learning techniques can work really well. At the same time, they're extremely powerful, and the ability to ingest a number of data streams, invite new areas of inquiry by learning what we can from them, and thinking about them, using them as a test bed to explore existing data and think how we can do that better. And while doing that in tandem, I think as we continue collaboration and strengthen our ability to work with experimentalists and observationalists in the field across different trophic scales, et cetera, we'll be able to improve our understanding of how we can parameterize models. And so just as I think experimentation and observations inform models, it can be the other way around as well. If there are certain parameters that we know are highly uncertain and influence models substantially, we need to call up our friends who are working in that area and talk to them about that and understand better what data do we need in order to constrain that. And while we have instantaneous snapshots in time as a problem, we do have SWOT. And so SWOT and PACE are best friends flying in space right now. And with SWOT data, we have incredible ability to see the submesoscale. And I think a lot of, when possible, when they overlap, a lot of interesting science can be done by combining them in creative ways and thinking about how these physical features can structure different communities. That we're showing chlorophyll here. This is from the Science Visualization Studio, but you can think about it as different. We could easily, put in phytoplankton communities and see how those are structured as well. And you could think about overlying, acoustic data, genomic data, et cetera. And then, asking questions about the ocean's evolution through time and how physical features structure. Why am I showing land? The reason why is I kind of wanted to close with an additional vision. I think that we can go now that we have PACE flying, and this is PACE, and it's incredible ability to let us see land in new ways. I also love this because it looks like a geological map, but we have this intuition, and we've been inheriting this, I think, since probably we were children in classrooms, seeing maps and understanding intuitively a desert colored in tan is less productive than the Amazon, that's often like a dark green color on a map. And there are different ways we can think about how life is structured on land and in the ocean. But when you go to the ocean, this is a product from Nat Geo, and I look at this and see like an opportunity to fill in the ocean colors, if and that could be something that could be done in a hack week, for example, or in this type of conceptualization. We, I think, have a long ways to go, which will be great in thinking how we can better understand our ocean ecosystem beyond chlorophyll, and thinking about not only what phytoplankton are existing there, but how does their productivity scale and allow for this incredible diversity of life that we enjoy higher up to persist and thrive. I wanted to also make a case for slow and steady for hack weeks. And so I attended a hack week in 2020 with ISAT2 with this incredible team here. And we just got our software published this year. So we were a team of scientists who were primarily MATLAB users, and we really wanted to learn Python. Well, some of us more than others, but we knew we had to, I should say, because it offers so much. And we met once a week for an hour. We pretty much never worked outside of that time, and we developed some software that is, meant to make it easier for those who want to extract vertical information in the ocean. And so we have ISAT2, which allows for vertical profiles of photons, which can be converted to geophysical quantities like backscattering and attenuation, and also Argo, which offers a lot more information in more depth in the water column. But we kept at it, and we did publish some software, and we're still working on it and developing it as an open source tool for the community. And so whatever you're working on now and that you might like fall in love with your project, it doesn't have to be finished this week. It can continue, and it'd be great if it does, and you keep us posted and in the loop on how things evolve and how we can support you. I wanted to end with this question. It doesn't necessarily need an answer now, but just for us to think about, because I'm also thinking about this, and I don't have one answer. There are a lot of responses I can think of, but we have this incredible sensor in orbit, and we at the same time have historical time series data for the last few decades. And so what can we do now that we couldn't do, before February last year, to discover, quantify, and monitor biodiversity? Okay, I think it just about came up on time, so I'll end there. But thank you so much, everyone, and I'll take any questions.
Have to run around the room with a mic, but we definitely have time for questions, folks. Thank you so much, Kelsey. Do I have any questions?
Thank you. Sorry, I just learned that my video wasn't on the whole time, but you can see me now, and you'll see me tomorrow and Wednesday at least, for better or for worse.
Hi, Kelsey. Thank you for an amazing presentation. I really enjoyed it. I was just wondering if you could talk more about using LiDAR to enhance GIOP-based inversion. That really caught my attention. Thank you.
Thank you. Yeah, so the idea with that project, and you could also ask Jeremy and Sasha, I think, who are in the room, who are working on that with me, was to think, we have an inversion framework. We have, a poorly posed problem in the sense that we have remote sensing reflectance, one equation, and two unknowns of absorption and scattering. So the thought was, if we can fill one of the unknowns, scattering, with a known product, then it would help improve performance, if and only if we're seeding it with something that's actually high performance. So that was the inspiration for the work. It ended up taking us a lot of different directions, starting with sort of a confirmation exploration of backscattering globally and how it performed, where we learned that the LiDAR products available right now perform a lot better than I thought they did when I started working with them, which I expected they would perform kind of poorly, mostly because we're using LiDAR data from the Calypso mission, which carried the Calliope instrument. It's been since decommissioned, but there's great data spanning from 2006 to 2017, and I think the new reprocessing up to 2022 soon. But we found for relative errors with backscattering, I think of 17% or something. And when we did the same analysis with most aqua, we found errors that were a bit higher in the 20s. And so that gave us some confidence. But, it was a really simple analytical expression. And so we directly substituted the unknown backscattering to be solved with LiDAR data. And it wasn't without assumptions, right? So LiDAR product gave us backscattering at 532 nanometers. But in order to run the GIOP, you need spectral backscattering. And so we had to assume a spectral slope. And we did, we played out with sensitivity analyses, what if we choose a spectral slope of 1 versus a dynamically varying slope, et cetera. I think there's more work that could be done there, possibly with LiDAR data, but I think absolutely with other data streams. And as I mentioned, perhaps even using the PACE data themselves, but incorporating information like the time varying derivative of remote sensing reflectance, and asking questions about what does that mean? How do we use that best to squeeze out as much as we can from this incredible sensor? And if you're interested in I can send you the paper as well if you're interested in reading more about how that's done, and I can send you the code. It's delightfully easy. It's just substituting directly the backscattering, so it's pretty nice to run in that regard.
Kelsey, I don't know if you're still on our Slack, but if you post a paper or something like that in our PACE Hack Week Slack, you are probably still there from last year. We'd see it. Cool.
Yeah.
there's somebody in this room, actually I think it was the person who asked the last question, who yesterday was showing me images of really striking diurnal patterns in phytoplankton moving, well, maybe it wasn't a phytoplankton, but it was like chlorophyll intensity being, very diurnal. And I imagine this was in a freshwater system, but I imagine that may be a more common and than we may realize. And I wonder if you've kind of run across this or thought about these kind of diurnal patterns and how that affects these metrics that we're coming up with for biodiversity, because since our sensors go over a region at a particular time.
Mm-hmm. Thank you. Yeah, it's a great question. I guess I've thought about it a bit in two ways. The first is physiological, and so thinking about diurnal changes purely due to cell growth and respiration as the day goes on, which can adjust chlorophyll concentration quite a bit. And the other way is sort of implied, but the grazing side. So we don't see it. We might not be able to explicitly quantify it, and that's where it's implicit. But we can have changes in chlorophyll diurnally because there can be sometimes a really large signal of this diel vertical migration in different areas. And there's some different areas of thought, and I haven't seen enough evidence in either way to say this confidently. But maybe I'll ask it as a curiosity, as a question. When we have particles in the ocean in the mixed layer, does that create like a snack bar, for zooplankton? So the idea is that where you have these different density layers, are there, different particles that are kind of resting there that then zooplankton might come up and chomp on, and then they don't have to go higher up in the water column, et cetera. And that might also cause some of this vertical structuring diurnally, depending on, again, how strong the signal is. We do think that diel vertical migration is more prominent in clear waters, which makes sense if you think about it. In really turbid waters, the photons are going to be absorbed really early in the water column. And so if there's zooplankton or fish that are trying to avoid predation, they don't have to go that deep to do so before it's dark and come back. But there's so much, I think, that we can address from this. And, Mike Barenfeld published his diel vertical migration paper in 2019, and there really haven't been any follow-ups. And I think that there's so much that could be done looking at cross-regional differences, comparing more with acoustic data, et cetera. But it's a really interesting question, and I'd love to hear more from those if anyone's working on that for their project or just outside of the Hack Week framework. As we move toward an era where we're hoping to have an ocean optimized LiDAR in orbit within the next 10 years, understanding how to interpret that signal is obviously really key if we want to use it well. So knowing all the different processes that could work to force or not different biodiversity across 24 hours would be great.
Thank you so much, Kelsey. We are really looking forward to asking more questions of you when we bump into you in person. Thank you very much. And we'll
Thank you.
move on.
Thank
Thank you.
Tomorrow.